In the last video, we talked about variant calling. After this step is completed, you can use the effect prediction application based on SNP tool to annotate the variants and predict the effects that they will produce on genes, for example, amino acid changes, functionality changes, and so on. The report is attached to the generated variants file. In order to open it, go to Variants with Predicted Effects for Clark folder, located in the tutorial folder. Choose the NumBulgen file, go to Explore and View Report. The report contains the following items. Summary contains tool version, number of variants, number of effects, change rate, and other information. Change rate details table shows the length, changes, and change rate for each chromosome and patch. Number of changes per type table summarizes the number of variants as well as number of single and multiple nucleotide polymorphisms, insertions, deletions, and mixed variants. SNPs are the most common sequence variations in the human genome, and in a case of an NumBulgen enriched sample, 555K SNPs were detected. The next table shows the count and percentage of variants that have high, moderate, or low impact or are tagged as modifiers. High-impact mutations are significant changes to the DNA code. For example, frameshift mutations, nonsense mutations, deletions of more than 1% of chromosome, or even deletions of the entire exon, and so on. Variants with low impact are the ones that will not influence the structure or function of the encoded protein. For example, synonymous mutations or silent mutations. Moderate variants do not significantly affect the protein structure, but might change its function, and these may, for example, be missense mutations, codon deletions, or insertions. Modifiers are mutations in introns, intergenic, intragenic, or other non-coding regions. In the case of these samples, we can see that only about 0.04% of all annotated variants have high impact. 97% of variants are modifiers, and we can see the same percentage of modifiers in whole exome and whole genome sequencing samples. The next table lists the count and percentage of missense, nonsense, and silent mutations. The number of effects by type and region table lists how many variants of each type, for example codon deletions, are there for each genome, for example a particular exon. The variations histogram illustrates what regions of the genome are most affected by the mutations. In this case is the introns, with 60% of variants located there. Quality histogram illustrates the distribution of quality scores for each detected variant. The application also reports the coverage for detected variants. Next, insertions and deletions length histogram shows size distribution of detected indels. Next report is the base changes table that reports how many and what type of SNPs were detected. The transitions transversions table reports the number of their ratio in SNPs and all other variants. The frequency of alleles histogram shows how many times an allele appears once, being a singleton, or twice, being a doubleton, and so on. The codon changes table illustrates how many and which codons have been replaced. The next amino acid changes table shows how many and which amino acids have been replaced. A row indicates a reference amino acid, a column, changed amino acid. Last but not least, the changes by chromosome graphs show the number of variants per 10,000 kilobytes throughout the whole chromosome length. Such histogram is generated for each chromosome and patch present in the reference genome. This is the end of this video. In the next one, we'll talk about variant prioritization and exploring the variants in the Variant Explorer app. For more tutorials, visit the Genestack webpage or our YouTube channel.